Before I introduce the last speaker, let's thank the staff who's done a great job. So our next speaker is Alison Etheridge, who will talk about the motion of hybrid zones and how to stop them.
Hopefully that works. Great, thank you very much. So the area that I work in is mathematical population genetics. And the aim is to understand how the different forces acting on populations like spatial structure, natural selection, something we call random genetic drift, which is just randomness due to reproduction in a population, how have those forces shaped the patterns of genetic variation that we see in the world around us? Or putting that on its head, if I look at patterns of genetic variation in a species now, what does that tell me about how they arrived, about the history of that, the natural history of that species? Now, the way that geneticists actually are able to reconstruct information, we've only got a snapshot of a real population. We can do experimental evolution and we can get sort of time series, but in a natural population, typically we've only got a single snapshot, a moment in time when we're looking at the data. And so the way that geneticists try to understand the history is by looking at what are called neutral genetic markers. And those neutral sites, the neutral mutation rate, which tells us where the variability comes from. Without mutation, we wouldn't see anything. Everything would look the same. The neutral mutation rate is typically very, very small. But we're only able to observe things that have evolved over the timescales of those neutral mutation rates. So for the sort of problems I'm looking at, I'm interested in very large temporal scales corresponding to times when you're going to see a decent number of neutral mutations arising. So you'll see differences between individuals in your population. But also that then translates into large spatial scales. So I'm going to do things which look pretty crude. I'm going to write down a model which looks completely crude, and then I'm going to apply a scaling to it, which makes it look completely unrealistic. But it actually does capture some of what happens in the natural population. And I'm just going to illustrate with one specific example, which is the motion of hybrid zones, because that's a fairly easy one to explain without doing too much biology. And then at the end, I will throw down a mathematical challenge for you, which I've been working on and off for a while, and I have no good ideas, so I hope someone else does. Okay, so there we go. So what's a hybrid zone? A hybrid zone is a narrow geographic region where on one side I've got one version of a population, on the other side I've got a second population, and those populations are sufficiently similar that they can interbreed, but the hybrids are, for some reason, selectively disadvantaged. Now, there are lots of reasons that might happen. We're going to focus on it being because of their genetic makeup, because their genetics is forcing them to be less advantageous. It could be because of a change in the environment, and we'll see an example of that a little bit later on. And a particular population that got me interested in this is this population. In the UK, we call these snapdragons, but these are antirrhinum growing in the wild. This is where they grow. This is the Pyrenees between France and Spain. And my friend Nick Barton, who's an evolutionary geneticist at IST Austria, has the enviable task of going out to the Pyrenees every summer and picking flowers. When I first met him, he was working on grasshoppers, but they moved too quickly. Now he's a bit older. So we have flowers instead. And the hybrid zone, which is in this region, on one side, one of the populations has just pure yellow flowers, and the other purebred population has these pure magenta flowers. And then across the hybrid zone, which is a narrow region in between the two, we see these varieties of flower colors, more like the things that we grow in our backyards. Now, it's believed that for this particular population, the reason there's a hybrid zone is that the pollinators, so the bees and other insects, are better at recognizing yellow flowers and magenta flowers than they are at recognizing these intermediate shades. And that's believed to be what's going on. But it makes it, therefore, at least indirectly, a genetic effect. Because if I'm of mixed genetic type and producing flowers of this sort, I'm going to be less reproductively successful. Okay, so I'm going to write down a crude model of how that works, but let me just emphasize that hybrid zones are absolutely ubiquitous. We see them everywhere. And the way that a lot of the hybrid zones we see in Northern Europe between plant species arose it is believed, was because the last glacial maximum, which was about 18,000 years ago, the plants which had previously been living in Northern Europe were pushed into refugia, into places where it was still possible to survive. And while in those refugia, they became genetically distinct, but not so distinct that they couldn't interbreed. And then when the ice melted, they very, very rapidly came back together. And when you got to the contact zone where the populations met, that's where you get these hybrid zones. And the hybrid zones that we're interested in actually move around with time. And that's what we're going to be concerned with. Okay. So here's a very, very crude mathematical model, but it actually captures more of the biology than you might imagine. So we're going to focus on selection maintaining the hybrid zone, and it's selection against being of mixed type. Now, looking at those flower types, these are all the flower types, essentially. They're a very small number of them. It's not hard to believe that the flower type is determined by a small number of genes, and indeed that's true. And we're going to caricature that by just supposing that there's one gene, and it comes in two distinct types, little a and capital A. And if you carry two little a's, so each flower is going to carry two copies of each gene. If you carry two little a's, you're going to be yellow. And if you carry two capital A's, you're going to be magenta. And little a, capital A, something in between. Okay, that's going to be our model. Now, that's what that says. We're also going to suppose what are called Hardy-Weinberg proportions. This isn't a huge assumption. If selection on my population isn't too big, what this says is that if I look at the proportion of little a alleles, so allele just means different genetic types, right? So our alleles are little a and capital A. And if I look in my population, eventually we'll say locally, but for the time being, let's suppose there's no space. And I ask what proportion of the alleles are little a, so that's counting 2 for every yellow flower and 1 for every mixed flower. What proportion of them are, What proportion of them are little a, and I call that W bar, then the proportion of yellow flowers is W bar squared and so on. So in other words, the proportion of flowers of different types is chosen just by random sampling from a pool of alleles in which we have a proportion W bar of little a. That's all these Hardy-Weinberg proportions say. And in fact, if you're not in, it's called Hardy-Weinberg equilibrium. If you're not in Hardy-Weinberg equilibrium and you do one round of reproduction and there's no selection, you get back to Hardy-Weinberg equilibrium. So it's really not a huge assumption, but it's one that is very convenient for what we're doing. Now, to model the selection against heterozygosity, we're going to suppose that the relative fitnesses of yellow flowers, purple flowers, and mixed flowers are 1 to 1 to 1 minus s, where s is some small constant, okay? Now, it's a meaningless statement until I tell you what relative fitness means. And I'm going to tell you what relative fitness means by talking about reproduction, by its influence on reproduction. Whenever I introduce a constant, it's positive. I'm not good at negative signs, okay? So any constant I introduce, just assume it's positive if I forget to say it. And typically small as well. And that smallness comes back to this idea that we're interested in forces that we're observing over very, very long timescales. So actually what we can observe about things like natural selection in a natural population is really weak selection. It's things that have evolved over a long time. Okay, so someone who knows a lot of biology in the audience, so I'm not very comfortable about this particular description of reproduction, but it will do. So the idea is that during reproduction, each heterozygote plant, so each mixed plant, produces 1 minus s times as many germ cells, so those are cells of the same genotype as the parent, as the homozygotes, so the yellow flowers or the purple flowers. But in fact, each plant produces an effectively infinite number of germ cells, and they split into gametes, and gametes contain just 1 copy of each gene. So from those gametes, we produce a pool. So that's effectively the male and females, right, for your reproduction. And the next generation is formed by sampling 2 gametes from that pool, just uniformly at random. Okay. So where does selection come in? Well, if the proportion of type A in my adult population was W bar, then I can just work out what the proportion of type little a is going to be in my pool of gametes. W bar squared, well, everybody who's yellow, all the yellow flowers gametes are little a, so that's a whole W bar squared. For a heterozygote, remember we're producing 1 minus S times as many germ cells. Those germ cells split into how many heterozygotes are there? Well, there are twice W bar, 1 minus W bar, but only half of their alleles are little a. So that's what's on top. And then on the bottom is just the total number of gametes in the pool. And so we obtain this ratio. Now, I said that selection needs to be weak because we're observing things over very long timescales. So we're only interested in things which are evolving over long timescales. So selection is going to be weak so that S is small. And just rearranging the equation, just using geometric expansion, we find that the proportion of little a alleles in this pool of gametes is what we started with plus a correction of order S. And we're going to throw away terms of order S squared. And that correction of order S takes this form, and that's just algebra. So in particular, the change in proportion between the parents and the offspring is given by this expression here, okay? Right, so how are we going to use that? Well, first I'm going to take a deterministic approximation. That's just what we had on the previous slide. If my population of plants is infinite, then sampling to produce all the offspring is going to exactly replicate the proportion of little a alleles that are sitting in that pool of gametes, right? And so, and remember that we said that S was small and that we're interested in a large number of generations. So M generations here, that's corresponding to a timescale over which I can expect to see order 1 of these neutral mutations. So I'm going to see some pattern in my genetic data. And if M is large and S is alpha over M, well, that says that if I look at the increment over a single generation and I normalize it to approximate a derivative, I'm going to get an expression like this. And that sort of says, I mean, that I could approximate the way that the proportion of little a alleles evolves in these units of M generations, where M is very large. I could approximate it by this nice ordinary differential equation. But I'm really interested in spatial populations, so I'm going to add some dispersal. And if I add dispersal, I arrive at this equation, which is a very familiar equation to anyone interested in PDE. It's a special form of what's called the Allen-Kahn equation. Now, I should tell you, there is a mathematically principled way of arriving at this model, which starts with individual-based reproduction based on plants, and then we pass to a scaling limit. But it would take an hour to go through that. And so I hope that this is enough to convince you that we should expect some sort of dispersal term, and we should expect a nonlinearity of this sort of form for the way that the proportion of little a alleles is evolving. And notice that if W is bigger than a half, so if I'm in a region which is dominated by little a's, I get pushed upwards. And if I'm in a region dominated by capital A's, I get pushed downwards. And that will determine the behavior of this equation. We're getting there. So very importantly, there are finite population effects. So what we assumed to arrive at a differential equation was that the proportion of little a alleles that we saw among offspring precisely replicated what we saw in the pool of gametes. But in general, that's not going to be true, because what we're actually doing, even if we take this very crude model in which we're randomly sampling 2 gametes from that infinite pool to create the offspring, what we're actually seeing is a binomial distribution, right? So if we've got a population of size n, and we need 2n gametes, then the number of type little a that are actually represented in our population is going to be binomial with 2n trials and success probability W star. And so the expected number of type A gametes among offspring is indeed going to be given by this W bar star, which we defined before, which is here, right? So the expected proportion, so binomial, the expected value is twice n W star, and I divide by twice n to get back to a proportion, and that gives me back W star. But there's going to be some variability. And if you teach first year probability enough, you know that the variance of binomial is, well, twice n, because we've got twice n trials times p times q, so W star 1 minus W star. And if I'm interested in proportions, I better find the variance of the proportion is going to have a 1 over twice n all squared in front of that. And that gives me a term like this, which is really what we started with plus an order S over N correction. And S we said was small, and so this term is going to be negligible. And what that means is that actually in the absence of space, at least if the units of time over which I'm measuring the number of generations which I'm taking as a unit of time scales in the right way with respect to the population size, then I would arrive at a stochastic differential equation. And don't worry if you don't know about SDEs. I'm sure everybody here does because you have a broader education than we offer in the UK, but this will be the first and last time you see one. But what we would like to do, of course, is add noise, add space to that, add spatial motion to that. And morally, what we would do, so I should emphasize that, of course, this is the term that we saw before. And this stochastic term comes entirely from the fact that we've got a finite population effect. We're sampling individuals at random. It's random reproduction of individuals in a finite population. And morally, we'd like to just add some spatial motion onto here. But there's a bit of a difficulty with this, which is that this stochastic PDE, where this is what we call a space-time white noise, which you could loosely think of as an independent Brownian motion as every point in space, has no solution in dimensions bigger or equal to 2. One dimension, it's got a solution, and we can analyze it, but it has no solution in dimensions bigger or equal to 2. And later on, we're going to see that actually it makes a crucial difference whether we're studying populations in one dimension or higher dimensions. So that's a bit of a problem, but we can modify the approach. And Nick Barton and I, a few years ago, introduced an approach to spatial populations which allows us to capture this finiteness in, again, a mathematically principled way. But again, that's another hour's lecture. So I'm going to skip over that, but I'll tell you the effect of it at the point where we use it. Okay, so let's continue with our Allen-Kahn equation, because it's a very beautiful equation. So here's the equation we've just derived for the motion of our, for the proportion of little alleles plus noise, as we've said. And we'll start by analyzing it without noise and then see what happens when we add noise, which was actually our original, when we started work on this. And we is Nick Freeman, Sarah Pennington, and myself. So Nick and Sarah were both students of mine in Oxford. What we were originally interested in was understanding the effect of noise on hybrid zones. But we started with this deterministic equation. And I just want to show you another couple of hybrid zones. This is the mouse hybrid zone, mus musculus versus mus domesticus. And it runs down through Denmark, runs through Germany near Plön, which is near Hamburg, and comes down and meets the sea just east of Trieste. And this is hand-drawn, but you can see that the hybrid zone between mus musculus and mus domesticus, which look a little bit different from each other, so they've just got slightly different fur colors, it is a long, rather smooth hybrid zone, but it's very narrow compared to the range of these mice, which spread right across Europe. Okay, so that's one sort of hybrid zone, and that one is maintained by selection. It turns out that, I think it's, correct me, David, but the male hybrid is sterile or something, right? Yeah. So that is maintained by selection, rather strong selection in that case. But it moves. But it doesn't move very much, and we'll see why it doesn't move very much as we go on. This one doesn't move. This is another hybrid zone. This is the hybrid zone between Bombina bombina and Bombina variegata, so yellow-bellied toads and fire-bellied toads. And this hybrid zone is really wacky, right? Again, it runs all the way down through Europe, but that's much more interesting. But it's actually maintained by a change in the environment. So it turns out that one of these, and I always get it the wrong way around, I think fire-bellied is on that side. The yellow-bellied toad likes to lay its spawn in ephemeral bits of water that dry up in the winter, puddles, basically. Whereas the fire-bellied toad likes a little bit more predictability in its life and goes for lakes and rivers and bigger bodies of water. And if my geography were better, I would be able to point out mountain ranges that actually give you the kind of runoff difference between these. So you can actually recover quite a lot of the geography of Europe from these toads. But this is a very different hybrid zone. This isn't moving because, or at least only on the scale of tectonic changes, because it's maintained by the environment. But we're interested in these zones maintained by selection, like our antirhinum. Okay. So how are we going to study them? Well, what I want to understand is exactly what David just asked. I mean, are these zones moving? And because we're interested in these very large spatial and temporal scales, I'm going to zoom out, because you could imagine this is actually drawn in by Steve Carr with a thick pen. This is almost a one-dimensional interface, right? It's a little bit wider, but it's almost one-dimensional. It's of the order of tens of kilometers, whereas the whole of Europe is a bit bigger than that. Not as big as the US, but bigger than that. So I'm going to apply a diffusive rescaling to be looking over very large timescales and correspondingly very large spatial scales. So assuming that things are moving in a kind of symmetric way, a Gaussian distribution, it's natural to apply a diffusive scaling. So that's what we do. And if I apply a diffusive scaling, what's going to happen to my Allen-Kahn equation? Well, the Laplacian term's not going to change, right? That's what we mean by a diffusive scaling. But the nonlinearity is going to pick up a factor of 1 on epsilon squared. So it's going to have a much stronger effect. And remember, we said that if W was less than a half, this would be pushed towards 0. This is a negative drift. And if W is bigger than a half, we're being pushed upwards. It's positive drift. So what we can predict is that if epsilon gets very, very small, the solution to this equation is going to converge to an interface, so the indicator function of a set, at least if the initial condition is sufficiently regular. And for convenience, I'm just going to take m equals 2 and alpha equals 1, because that's where all the math has been done. Of course, I could scale that away. And in fact, what happens to that region is that it's not static. One's first guess might be it'd be static, right? Because we've got this incredibly strong force pushing us to be either 0 or 1. But this Laplacian is enough to shift it. And the way that it shifts it is we get convergence to the indicator function of a region whose boundary evolves according to something called curvature flow. Now, I imagine, again, that you well-educated people will all know about curvature flow. In higher dimensions, it converges to mean curvature flow. I'm just going to say curvature flow because, of course, in 2 dimensions, it doesn't matter. All the maths that I talk about works perfectly well in any dimension. Our anti-Rhinum live in 2 dimensions. So the slides will focus on 2 dimensions. Occasionally I'll forget and there'll be a D, but if there's a D, it means D equals 2. Okay. So curvature flow, just in case, but as I'm sure you all know, but let me remind you. So here is the boundary of my region, right? I've got a nice enclosed region. So I'm supposing that yellow flowers dominate in here and purple flowers dominate out there. I can parameterize that boundary, I'm supposing everything's smooth enough for everything I say to make sense, by the way. I can parameterize that by the unit circle, right? And I'm then going to allow it to evolve. So at value u in S1, I'm going to write NT for the unit inward normal to this, where I'm supposing this is wherever the boundary is at time t. So if this is the point u, this is the inward normal going this way. And I'm going to write kappa for the curvature of that curve at u. So that's 1 over the radius of the osculating circle, if you like. So the best fit circle, and I look at 1 over the radius of it. And curvature flow, well, the way that the point gamma t of u moves is that it moves along the inward normal at a rate proportional to the curvature. So this point moves faster than this point, and this point's going to move inwards, and that point's going to move outwards. And there's an exceptionally beautiful theory of curvature flow that was developed in the late 80s, really, by people like Gage and Hamilton and Grayson, which says that in 2 dimensions, it's slightly more complicated in 3 dimensions and higher, but in 2 dimensions, after a finite time, this curve is going to evolve into something which encloses a convex body, and asymptotically, it's going to, it'll shrink to a point, and it'll become more and more circular as it does so. And of course, it'll hit that point in finite time, because if you imagine starting from a circle, you're moving at a rate 1 over r, and so as the circle gets smaller, you go faster, and you actually hit 0 in finite time. So this curvature flow is only defined up to some fixed time, which I've denoted by t. Okay, so with apologies to the geometers. So far I've offended the biologists by doing bad biology. Apologies to the geometers for doing bad geometry. But yeah. So in fact, the reason that the Allen-Kahn equation was originally developed as the Fitz-Nagumo equation and curvature flow coming out as a limit was coming from physics, that's right. Okay, so what does that? I made the statement that the solution to this equation as epsilon went to 0 would converge to the indicator function of a region whose boundary evolved according to curvature flow. Can we state that a bit more rigorously? Well, we can if we take a nice enough initial condition. So assume I've made my initial condition nice enough, please. I'm not going to write down the conditions, but they're not too bad. And what we're going to suppose is that gamma 0, which is the set where our initial condition takes the value 1/2, is just a nice curve, right? And then as time, what other assumption I'm making? I'm going to assume that inside this region, inside gamma 0, W0 is bigger than 1/2, and outside it's less than 1/2. Everything's symmetric, so I could say that the other way around, but that'll do. All right. And then the statement is, roughly, that if I look at any time before that time when my curvature flow is no longer well defined, then at least for epsilon small enough, if I allow the solution to evolve for a little bit to get the hang of what, to settle down to something well-behaved, then if I'm far enough away from the boundary, which is now evolving according to curvature flow on the inside, then the solution to my equation is arbitrarily close to 1. It's bigger than 1 minus epsilon to the k. And if I'm far enough away on the outside, my solution is very close to 0. It's less than epsilon to the k. So for any epsilon, I can find an epsilon of k so that for all epsilon less than an epsilon of k, at least if I let the solution evolve for a little bit, then inside here, a distance at least epsilon mod log epsilon, I'm bigger than 1 minus epsilon to the k. Outside, a distance at least epsilon mod log epsilon, I'm less than epsilon to the k. So in that sense, I'm very close to being the indicator function of a region. Now, we got very excited when we saw this result because we thought, oh, now that's that sorted. We understand hybrid zones. All we've got to do to understand the effect of noise is to look at Chen's proof and modify it a bit, right? And that'll be great. And we'll be able to understand what noise does. But unfortunately, although Chen's proof is extremely beautiful, it's also rather inflexible. And we discovered that we could not just adapt it to add some noise, not least because of that issue that adding noise in 2 spatial dimensions requires a bit more work than in 1 spatial dimension. It's not a totally local object. And so the first thing we had to do was come up with a probabilistic proof of Chen's result. And I think it's quite a nice proof. So I'm going to show you what it's based on. So what we do is we find a way of representing the solution to the Allen-Kahn equation probabilistically, so in terms of a stochastic process. And the stochastic process in question is something called ternary branching Brownian motion. So what do I mean by that? Well, we're always going to just start from a single individual. Think of this individual as being at the point x at time 0. And that individual is going to evolve through time according to a Brownian motion until it dies. And when will it die? Well, it dies after an exponentially distributed lifetime with parameter 1 over epsilon squared. So with mean epsilon squared. So as epsilon gets small, it really does not live very long. It lives a very short amount of time. And when it dies, it's replaced by 3 offspring in the location where it died. And there were 3 offspring here, but the lines overlapped. You can probably see here, we've got 3 different Brownian motions going off. And then at the fixed time in which I'm interested in my solution to the Allen-Kahn equation, so it's a time up here, I've got some finite number of particles in space. They're just at some finite number, random number, nt of particles, and they're at random points in space. And what we're going to need to record is both the positions of these leaves and their relationship through this tree. So what's the tree? You can make these lines straight lines instead of colored lines, but we need to understand the relationship between individuals in the tree. So we preserve the tree of relatives, and we preserve the locations of these leaves. Okay. Right. And there's a typo, this should be de Marzi. So, and then we adapt an idea of de Marzi, Ferrari, and Leibowitz. So what's the idea? So I'm writing W of t for that whole tree, that historical ternary branching Brownian motion. So the tree is swept out by my process. And then for my initial condition, W0, what I do is that wherever there's a leaf, I evaluate W0. And that gives me a number between 0 and 1. And I use that number as a probability in a coin tossing experiment. So my coin comes up heads with probability W0 evaluated at that leaf. And if it comes up heads, the leaf votes 1. And if it comes up tails, the leaf votes 0. Okay. And I do that at all my leaves. And then I trace back down through my tree. And whenever I get to a branch, the vote of the parent is going to be the majority vote of the offspring. Okay. And that gives me an iterative procedure, which takes me from the leaves down to the root. And I'm going to use this notation, VW0, to be the vote associated to the root. Okay. So a picture speaks 1,000 words. So this is just to remind us that all these lines are actually following Brownian motions. And at each of these leaves, I've done what I said I'd do. I've evaluated the initial condition at the point where that leaf is in space. I've flipped a coin whose probability of coming up heads is determined by the value of my initial condition at that point. And I've assigned 0 and 1s accordingly. And they're independent at each leaf. Okay. And now here, for example, 0, 0, 1. So I trace back. Here's my first branch point in this tree as I trace back from the leaf to the root. And 0, 0, 1, the majority is 0. So this branch picks up a 0. The next one's here, 1, 0, 1. The majority is a 1. So that's a 1. 1, 0, 1, there's another 1, and then 1, 0, 0, that's a 0. So down here, 0, 0, 1, so the root vote is 0. Okay, so it's just majority vote at each point. And we're going to consider this quantity, which is the probability that the root vote is equal to 1. If I start my ternary branching Brownian motion from a single individual at x at time 0. Okay. Now, to see what the connection is to Allen-Kahn equation, notice that the probability that I vote, that the parent votes 1, if for each of the children, we've got a branching process, everything is independent. So the probability that this child votes 1 is the same as the probability this one does or this one does. It's all completely symmetric. And so if the probability for one of those being a 1 is W, the probability that the majority vote is a 1 is W cubed plus 3W squared 1 minus W. I'm allowing at most one of them to vote the other way. And I can re-express that as the nonlinearity that's arising in my Allen-Kahn equation plus W, right? So the change in the probability of voting 1 as I go through a branch point is precisely the nonlinearity that I want from my Allen-Kahn equation. Now, I'm branching at rate 1 on epsilon squared, and so the rate at which I see that change is going to have a 1 on epsilon squared sitting in front of it. And the branches are all following Brownian motions, and that's going to give me a Laplacian. So that's where the Laplacian comes from. And it turns out then that the solution, this probability here, the probability the root vote is 1, is the solution to the equation in which we were interested. Okay. Now, it's a slightly complex process, but it turns out it elucidates rather nicely what's happening in this Allen-Kahn equation. So first of all, what's change result become? Change result becomes that the probability that I vote 1 is at least 1 minus epsilon to the k if I'm inside my domain, and the probability I vote 1 is at most epsilon to the k if I'm outside my domain, whereby outside I mean a distance at least epsilon mod log epsilon from the boundary. But what are the two mechanisms that are causing that to be true? Well, the first mechanism is this majority voting, because majority voting amplifies bias. If you're in a casino, they have a probability bigger than a half of winning any game. If they offer you best of 3, do not take it, because if their probability of winning was bigger than a half, their probability of winning at best of 3 is even bigger. You may as well just walk out after one game and lose your money right away. Okay? And similarly, if P is less than a half, it amplifies the bias. This quantity is less than P. So if we have lots and lots of rounds of majority voting, which is what we have in this process where we're voting at rate 1 on epsilon squared. So in order 1 at time, any particular route down the tree has seen on the order of 1 on epsilon squared branches. That's a lot of branches. So this amplification is going to take initial condition either very close to 0 or very close to 1. And the other effect we need to understand is that if we look at the distance between a two-dimensional Brownian motion, so that's what each branch of my tree is following, and the curve that's evolving according to curvature flow, my boundary, then I can couple, that was my notation for that distance, and I can couple that with a one-dimensional Brownian motion. And that uses some regularity on my initial condition, but is otherwise not too bad. And then just to take the heuristics a bit further to see why that's going to give motion according to mean curvature. Let's suppose that we've already generated an interface. So many rounds of majority voting is going to generate something that looks like an interface. And let's pretend, for the purpose of the heuristic, that this is actually a sharp interface. And let's also pretend that we have a circle, because it's easier to explain. Then what we've just said is, if I want to find where the boundary between voting 1 and voting 0 is, well, it corresponds to the point where the bias, which is just for each leaf, the probability that the leaf is outside the ball, is a half, right? Because out here we're 0, and inside the ball, we take the value 1. So where is it going to move to in delta t of time? So I'm going to move from this boundary at time 0 to some interior point x. And what is x going to be? Well, it's the point where the probability starting from x, a two-dimensional Brownian motion has probability a half of being outside the ball at time delta t. But a two-dimensional Brownian motion is a one-dimensional Brownian motion with a drift of order 1 over its modulus. And if I'm close to the boundary of this ball, and the radius of this ball is capital R, then 1 over the modulus of my Brownian motion is 1 over R to a first approximation. And so actually the modulus of W delta t is essentially B delta t plus 1 over R times delta t, plus something lower order, which I'm going to ignore. And we're asking for the point x so that the probability that that is bigger than R, so that W lies outside the ball, is equal to a half. But then I just rearrange this, right? And then that's the probability B delta t is bigger than R minus 1 over R delta t. And one-dimensional Brownian motion is just symmetric about the origin. And so that says I must choose x to be R minus 1 over R times delta t. And that's saying I move in according to the curvature of that circle, 1 over R. So that's where the curvature flow comes from in this argument. And I think it's rather nicely elucidated by the stochastic representation. And that can be compared actually to something called the MBO algorithm, which is used for numerical generation of motion by mean curvature. Okay, what comes next? All right, so far, we've supposed that our yellow flowers and our purple flowers are equally fit. But in fact, there's no particular reason for that. So well, before I say that, so yes, my mouse hybrid zone is moving. But what it's trying to be is, because it's going the whole of Europe, it's actually trying to be infinitely flat rather than shrinking to a point. So curvature flow for something like the mouse hybrid zone, which is stretching between the two the whole length of Europe, is going to essentially push you towards being a straight line. And remember that the mouse hybrid zone did look incredibly smooth. Right? It's been there for a long time, it's null species, and it's almost straight, in sharp contrast to this. So you do see a difference in these hybrid zones according to what's causing them. Okay, now, I was, of course, going to tell you what happens with noise, but I'm going to tell you what happens with noise in a slightly more general setting. Yeah, probably right. Okay, so far we've said yellow flowers, purple flowers are equally fit. But actually, if those populations have been evolving independently for some time, that may not be the case. It may very well be that the yellow flowers, these little a homozygotes, are fitter than the purple flowers. So let's consider a model where they're fitter than the purple flowers, but the purple flowers in turn are fitter than the heterozygotes. Okay, so there's going to be some bit of an advantage to being yellow instead of being purple. And there's plenty of reason to believe that this is something that's going to happen in hybrid zones. Well, the same derivation that I showed you before, or rather a sort of principal derivation based on individual-based models, leads to this modification of the model that we had before, which, if we rescale and we take the gamma to be small enough, can be expressed in this form. So this bit, W1 minus W twice W minus 1, looks like what we had before, but there's also a drift new W1 minus W times 1 over epsilon, so which is going to push things out, is going to cause the yellow population to grow. Okay. And in his thesis in 2018, Mitch Gooding showed that you can prove an analog of the Chen result for this equation, and indeed for this equation with noise, but we'll come to that in a minute. And the limit now is, instead of just being curvature flow, it's a mixture of curvature flow and constant flow. So, and those things push against one another. So curvature flow, remember, was pushing us down an inward normal. So in this picture, the curvature flow is pushing us downwards into the circle. And the constant flow, because these are the yellow plants, remember, is pushing us outwards. It's giving us an advantage. So minus nu, because NT was the inward normal. Okay. And again, that's defined up to a fixed time. And you can see that there's a balance. There's going to be a critical radius where this would exactly balance. The outwards push and the inwards push are exactly balanced. But it's rather artificial. You're never going to see it in nature because it only happens for exactly one radius. If you start from something smaller, you'll shrink to a point because the curvature flow will win. And if you start from something bigger, the constant flow will win. However, what we were really interested in was more realistic shapes of domain. Now, had we been working in one dimension where curvature flow doesn't come into play, then it's well known that this equation, so this is the same equation, exhibits traveling wave solutions. And for those who care about these things, it's what's called a pushed wave. And for the one person in the audience who cares about these things, that means the genealogy will be completely different from what you see in Fisher KPP. So what does this mean? Well, it means that I can, in fact, express the solution, or a solution, there is a solution, which can be expressed as a traveling wave of this form, which moves at speed minus nu. So that was the constant which measured how much fitter the yellow guys were than the purple guys. And it connects there being entirely purple flowers at minus infinity to entirely yellow flowers at plus infinity. And this picture was drawn using a Zoom whiteboard, and they don't have purple. So if the flowers aren't quite the right color, it's because I'm too primitive with technology. Okay, so that's what happens in one dimension. So in one dimension, the yellow flowers being fitter, they would invade completely. They would be able to completely take over the purple flowers, and at a finite speed, so we might still be able to see an interface. It would be being pushed out towards minus infinity. But very few populations live in one spatial dimension. So we thought, well, what about higher spatial dimensions, and two in particular? So when is it going to be possible for a population to invade in two spatial dimensions? Now, if my two spatial dimensions is all of R2, and I have a population which is occupying the half plane, say, so I've got a half plane of yellow and a half plane of purple, then obviously I can just project that onto one dimension and the yellow flowers will invade. But again, when you think of expanding populations, they're not really evolving on all of R2. And we were particularly interested in what would happen if they had to get through an isthmus, a narrow strip of land. It's quite a natural thing to happen to them, that they have to go through some interesting geography. And again, we found that the PDE guys had got there first at some level. So this equation with the initial condition, well, we're going to take a domain omega in R squared as it's for where it's living. So it's not the whole of R2, but it's some domain in R2, and we're supposing it contains the x-axis. So we're still looking at something infinite, but not necessarily infinite in all directions. And for initial condition, we're just supposing that every point to the right of the origin is occupied. So yellow flowers, as soon as your x coordinate is bigger than 0, you're yellow, okay? And in probability, we have to specify which Berestiki we mean, because this guy has two sons, both of whom are now rather distinguished probabilists. But this is Henri. And Henri Berestiki and friends proved a very lovely theorem, which is paraphrased to say, depending on the geometry of the domain, the yellow flowers can completely invade. They can partially invade by invading in maybe one direction, but not another direction. You can have it that they spread all the way out to infinity in one direction, but maybe something weird happens in another direction. Or you can have total blocking. The population cannot spread. And as I said, we were particularly interested in what would happen with an isthmus. So we focused, first of all, on a real caricature, a domain that looks like this. So we have a narrow cylinder, which feeds into a wider cylinder. So the yellow plants are coming along like this, and suddenly their domain opens out. Now, if little r0 were equal to capital R0, the yellow plants would simply invade, as we had before. But if little r0 is less than capital R0 and epsilon is small enough, then we can have what we call blocking. So there is a critical size, that D should be 2, because I've drawn this in 2 dimensions. If I can put in this case, a semicircle, of course, but hemispherical shell in general, around the opening, which has radius less than a critical value, then the yellow plants will propagate all the way along this narrow cylinder, but then here they'll get trapped. They won't be able to spread indefinitely. And what's happening, of course, is that when the domain opens out, their offspring are spread too thinly, and then they can't compete. Then the next generation, gene flow pushes them back. So you can think of it as a kind of surface tension-like effect, that curvature is going to be pushing them back into this narrow cylinder, and they can't proceed. Okay, and of course, we could take other domains. All that really matters is that we can put a portion of a spherical shell inside the domain of a radius less than this critical radius. Okay, so that says that the shape of the domain is really important because it can block things. But so far, we've only talked about partial differential equations. We've only talked about things that correspond to infinite populations. They're infinitely dense everywhere. But real populations aren't infinitely dense. Real populations have a finite density, and that effect of resampling that we saw at the beginning, the finite population effect, is going to matter. And so what's going to happen here? Well, again, I'm just writing down something which is morally true here rather than something that's actually true. But what happens when you introduce noise is that it acts against the selection. There's a mantra in population genetics that natural selection is most effective in bigger populations. The bigger the population, the more effective selection. And what happens here is that when we modify things to be able to write down a model which incorporates noise, instead of just having a structure corresponding to branching Brownian motion, we have something that corresponds to branching and coalescing. Now, it can't be Brownian motions because 2 Brownian motions in 2 dimensions don't meet, which is the root of all the problems of 2-dimensional population genetics. But we can modify the model in such a way that these lineages, these branches in our ternary trees, they follow a stochastic process, which is actually a random walk in our setting. And when they're close enough together, they have some probability of coalescing back into a single line. And every time they coalesce, that's effectively annulling a branch point. Because if 2 lines coalesce, then as I trace back from the leaf and do a voting mechanism as I do before, those 2 lines necessarily have the same type. So they will be the majority at the point when they're created. So if there's too much coalescence, it counteracts that selection. And that has the effect of changing epsilon, if you like, in our equation. And so it can break the blocking. The critical size of our isthmus can get broken down. So we have a branching and coalescing structure, and if there's too much coalescence, then it's going to break down this effect of the population being blocked. Now, too much coalescence actually corresponds, so the genetic drift, this is what drives the coalescence for reasons I'm not going to explain to you, but too much coalescence corresponds to this beta being very big. And beta is inversely proportional to the population size. When we wrote it down without space, it looked like something divided by n. So if the population density is low, this stochastic term, which we call genetic drift, is very high, and it breaks down the kind of surface tension that was holding back my population. But what's interesting is if the population density is high, we still get blocking. But if the population density is low and we get this surface tension broken down, what's actually going to happen is not that the yellow flowers invade, it's that we're going to see coexistence of types. So selection's being really ineffective, and the thing spreads across the whole domain. But the proof of this result, it really depends on this same branching system and voting idea that we had before, but now we have coalescence as well. So by virtue of taking a probabilistic proof of Chen's result, we were able to do something much more general and consider the effects of noise. Okay, there are other things you can do. You can do things like put long-range dispersal in and work out when you're still going to get blocking, you're still going to get motion by curvature flow. And there's all sorts of games you can play, and it's quite fun. But arguably, it's, we've sort of pushed this quite a long way. and one needs to think of the next challenge for it. So the conclusion of what we've done so far is that space matters. If I didn't have space, if I just looked at the stochastic differential equation without space, so suppose that Laplacian term was not there, then my population would ultimately fix on entirely little a's or entirely capital A's. So I'd have either entirely yellow flowers or entirely purple flowers. There'd be no concept of coexistence. So we wouldn't be capturing anything of what might be happening in space. So space matters. The shape of the domain matters, and that was something which hadn't really been taken into account very much before, but I think it's very important that we do. The dimension of the space matters, so one dimension is not enough. So just because we can do differential equations in one dimension, it doesn't mean that's the end of the story. And the stochastic term, the noise really matters. It really changes the behavior of the population. Okay, so where next? What would we do next? Well, with enough energy, you could extend this approach to traits determined by types at some small number of loci. This voting idea gives you representations of solutions to partial differential equations with polynomial nonlinearities of any order that you like. It becomes increasingly intractable to study, but there is now actually a literature developing from the PDE guys that's trying to do exactly that. But many of the traits in which we're interested in are not determined by a small number of genetic loci. Many of the traits which our forebears were studying at what was called the modern evolutionary synthesis, which was when mathematical or theoretical population biology really took off, or mathematical population genetics really took off. These were the likes of Fisher and Wright and Haldane and so on, and they were really interested in quantitative traits like height and weight, and more complicated things like how well we cope with the cold. And these are determined quite possibly, and certainly height and weight, are determined by an accumulation of very small effects at a very large number of loci, plus an environment. Of course, the environment in which we live matters. And traits that are involved, that are determined by a very large number of different genes, loci just means, just think of locus as meaning gene. Traits determined by a very large number of loci are not going to be things that we can capture using this approach. So we're only able to look at hybrid zones that are determined a bit like our antirhinum using this approach. But we need to understand these things, and we need to understand these things because populations are subject to change, right? And we want to know how they're going to respond to things like climate change. And the very simplest model we can write down is something called the infinitesimal model. And this goes back to Fisher in the first quarter of the last century. And Fisher's infinitesimal model just says that, well, my trait is determined by the sum of a lot of very small effects at different loci. You suppose that those effects are independent at different loci, except through the way in which individuals are related to one another. And we've used Fisher's infinitesimal model to try to understand what's called evolutionary rescue. So what's evolutionary rescue? Well, evolutionary rescue is when a population finds itself maladapted to its environment and needs to adapt quickly. And the way that it does that is because natural selection acts on the variability in the population. So just as natural selection works better in a big population than a small population, natural selection doesn't operate at all if there is no variability in the population. And so in order to adapt, you need to have variation. And Fisher's infinitesimal model gives us a very nice tool for understanding what happens to a population as it tries to adapt. And what happens is it loses a certain amount of its variability, typically, so that if the environment changes again, it's going to find it harder to adapt. And some rather counterintuitive things happen. So it's very important that we have a mathematical model that really explains this kind of adaptation to multiple loci. So let me just tell you one sort of counterintuitive thing that happens. It's not counterintuitive once you've understood it and written out the math, but I found it counterintuitive when I first heard it. So suppose I've got a population that's trying to adapt to living on an island to which it is not well adapted. And we've got some sort of stabilizing selection. And this population is being fed by migrants coming from a mainland, but the mainland migrants are not well adapted to this island. And what one wants to know is how fast do the migrants come from the mainland in order to optimize the chance of getting a well-adapted population established on that island. And the first guess would be that you want individuals to migrate as quickly as possible from the mainland. But that's wrong. If individuals may come too quickly from the mainland, you end up with a population which will be large, but maladapted, so that if the flow of individuals coming stopped, then it would probably die out. And the reason for that is that as a population starts to adapt, the individual here is fitter, but if it's breeding with a less fit individual from the mainland, it will get less fit through reproduction. So selection won't be able to counteract the rate at which the population is being dragged back by the mainland population. Now, I found that really quite counterintuitive. And so we've been trying to develop mathematical approaches to the infinitesimal model, which allow us to investigate these kinds of effects. And one of the reasons that's difficult is that the way that we've modeled a single locus, it was very easy. We just said, oh, well, we sample alleles from an infinite pool of gametes. Right? But if I'm actually trying to model lots and lots of loci, even if they're on different chromosomes, they're not independent of one another. We tend in population genetics anyway to think if you're on different chromosomes, you're evolving independently, but you're really not, because you're constrained by having the same parents. So the choice of parents, the pedigree, is something which is constraining Mendelian inheritance. And what we need is a mathematical model that doesn't just say, I've got this allele distribution moving forwards in time. We need a mathematical model that captures the fact that individuals in our population choose two parents, and the way that they then choose their alleles at different loci is constrained by the way those parents have been chosen. And that introduces something really very complicated into the model. Now, if you don't take any notice of the finite population effects, it doesn't matter. If you're interested only in infinite populations, you can still write down a differential equations model, which will allow you to investigate the evolution of the infinitesimal model with time. But finite population effects, especially if we're interested in adaptation to new, let's say, climatic conditions, as a random example taken from nowhere, of course, are going to be increasingly important. So what we need is a tractable, and I choose my words carefully, I can write down a mathematical model, but I can't write down a tractable mathematical model that keeps track of these so-called pedigree relationships, the way that individuals are related through parents and grandparents and great-grandparents, a branching and coalescing structure that goes backwards in time, and the trait values, which are determining their ability to adapt to a new environment. So that's the challenge for you, which I certainly don't know how to solve. So all ideas gratefully received. And it's time to stop. Thank you very much.
Thank you, Alison, for a great talk. Time for questions.
Thank you for indeed a great talk. You described this blocking coming out of an isthmus. Is it easier to block coming into an isthmus where the constriction narrows in the direction of?
You'll just move faster. If you've managed to get to that, you'll go straight in.
But there's
no blocking going in. You can't block going in. You can only block going out. It's because your offspring needs to be spreading thin enough so the hybrids are going to be. You need to be hybridizing enough for your offspring.
And then one last question, please. You've been talking about these beautiful hybrid zones in Europe. Of course, that's on land.
Yeah.
What about in the ocean? There can be phytoplankton, there can be cyanobacteria. What happens to hybrid zones when they're acted upon by some kind of moderate Reynolds number flow?
I do not know the answer to that question. That's an interesting question. I was hoping you were going to say, what about North American Audubon warblers or something? And I could say,
North America anyway?
Well, North American Audubon warblers are a classic example of hybrid zones. We don't know how they're maintained, but it is genetic, right? But I don't know about the ocean. I think that's a great question, actually.
Thanks. Great talk. I learned a lot. Let me ask a related question, which is like, I guess we'll take the infinitesimal model and it's just a neutral variation. And then it's presumably like a diffusion equation and you can just have a gradient. What would determine the size of that gradient? Would it have to be just?
Well, the problem that you have with the infinitesimal model is that So if I take two offspring, so exactly your neutral setting. If I take two parents and I look at their trait values, the offspring will have a trait value which is the mean of the parental value plus a normally distributed random variable. Now, the variance of that normally distributed random variable is given by something we call the probability of identity, which basically says if I sample a gene from here and a gene from here, what's the probability that they had a common ancestor back in the pedigree, that they came from a common ancestor back in the pedigree. And of course, as your pedigree becomes inbred. And so I can't really answer the question because it depends on the pedigree.
Okay, I'll tell you the problem. Maybe you've thought about it. There's this haplomap of human DNA variation, and there's these leading kind of factors, and you know what I mean. There was a famous paper around 2010 that did one of the early large-scale genome sequencing projects, and they discovered you can recover the map of Europe from the.
Oh yeah.
So there's a gradient in there. So I was.
Oh, well, the important thing there is that there is spatial structure. So what I just described didn't have the spatial structure. To be able to do that. So that, I mean, what's determining? So the way that they reconstruct, that's from colleagues of mine, Peter Donnelly and Simon Myers, were key players. There were many others involved too. Gil McVeigh, lots of others. Big project. They do a fine scale map of all of Europe. There's an independent project from Graham Coop and Kyan Davis. And basically they reconstruct the political history of Europe. It's quite extraordinary by looking at genetic relationships, but they're really reconstructing the map by looking at how closely related. And this is something, it's again the probability of identity, but the probability of identity when you're a spatially distributed population classically would be given by something called the Wright-Malakoff formula, which says that the probability that a gene in me and a gene in you is the same. Well, if you sample us from a distance x apart, it decays approximately exponentially. And essentially that's what they were using, was piecing together these maps. But you can do it in two ways. You can do it by looking at the individual loci and then trying to understand the correlations, or you can look at, and the HapMap project, what they did was they said, well, how long are the shared blocks of genome? So there are two ways. I've expressed it in terms of probability. A gene in me is the same as a gene in you. You could also say, well, if I take a block of genome, what's the probability that we share that whole block? Or how's the longest block? And that's determined by recombination. So you basically look down the two, necessarily as you go back in time, you'll be able to trace our genes back to a common ancestor. And it says, was there something called a recombination event? And again, recombination, it's a lot like mutation. And so you can use it as a clock. And it's again measuring, the length of the haplotype block tells us essentially how our ancestors moved apart.
Okay.
Does that answer your question?
Well, just to come back to the question, does it mean that there is a preferred gradient, or does it mean the gradient grows with time, or just look at one factor and one?
A gradient to what though? Gradient.
So we have some factor in this factor analysis of just the
oh right,
is the neutral variation.
Oh well, it'll grow with time, won't it? A single thing will start to. Oh wait, which way around? Yeah, a single thing will start to dominate.
It will grow.
Yeah.
Okay. Thanks.
Thank you for the fascinating talk. I was wondering on this open question here, are you thinking about additive accumulation of multiple loci, or are you thinking about more general functions? And is there any case you can analyze?
So the simplest model, the additive case, is the one that everybody considers. And there's a reason for that, because it's, well, okay, so you could do something slightly more complicated if you were willing to throw away the finite population size effect. But I'm not willing to throw away the finite population size effect, not only because I'm a probabilist and want to make probability sound important, but because actually, if we are trying to understand things like evolutionary rescue, we're looking at populations that are getting small. And if you look at populations in spatial distribution, they don't have infinite spatial distribution. If you're willing just to write down a differential equation, there are various approximations you could make, probably even in the non-additive case, though people don't do it. The additive case has the advantage that in order to understand what the trait values of offspring look like, All I care about is the trait values of the parents and this quantity called the probability of identity, which is just a second order statistic of the pedigree. If you want to understand more complicated things like dominance, so we actually have written a long paper on the infinitesimal model with dominance, then you actually need higher order statistics of that pedigree. Now, that pedigree is huge, typically. So what the pedigree measures, so if you want to understand my ancestry, you might think that you want to look at my parents, my grandparents, my great-grandparents, and so on. And of course, that grows very quickly. So before too long, you get to individuals that occur multiple times in my ancestral tree. So actually, it's not a tree, it's a branching and coalescing structure, but it's quite a big branching and coalescing structure, most of which won't have contributed very much to my genetics, in fact. Most individuals in my pedigree won't have passed any genes on to me, but we don't know that if from the information of the pedigree. We only, you only know it if you've actually got all the information about what ancestral genes look like. And so the way that you model it is this complicated branching and coalescing structure, and then run random walks along that structure, essentially. And if you want to understand more complicated things than the additive case of the infinitesimal model, you need third and fourth. I mean, even with simple dominance components, for example, you need third and fourth order identities. And with Nick Barton's MacBook and ability to do things in Mathematica, that's not on. We can't really even model the fourth order identities. It's one of the things we want to be able to do, but we don't really know how to do it. But the infinitesimal model breaks down quite quickly, but on the other hand, it seems to be, it's a surprisingly good model for what goes on in the background. So what there's a lot of interest in doing is stripping out genes that have a big effect. And in fact, animal breeders have been doing this for decades. You look at things like, if you're a dairy cow, there are 1 or 2 genes that make a really big difference to how much milk they produce. And then in the background, there's something that looks like this additive trait. And so the idea is strip out the effect of the big genes and look at the rest as an additive trait. And it seems to be quite a good model.
Oh, thank you.